Hi, I'm Catherine Perez, and I'm at the University of Texas, Rio Grande Valley. Thank you for coming to my talk. I want to tell you about some of the work that I've been doing along with my students and collaborators, Benjamin Schwartz and Benjamin Hutchins at Texas State University. We've been working for the last few years on the subterranean gastropods that are found in the Edwards Trinity Aquifer System in Texas and Mexico. On the right here, you see Balcanornis coronae, which is a pretty typical size for this group of snails. So they're all very small. Now, some of the agencies that have funded this work include Texas Parks and Wildlife through a state wildlife grant and the U.S. Army Corps of Engineers through a much larger project that's looking at river and hyporheic dynamics. We've also had some funding from the City of Austin and Williamson County, as well as much assistance from UTRGV. Now, aside from funding, this work would not be possible without many people's assistance, such as people giving us access to land, museum collections, people helping us take samples often in inhospitable and remote environments, and then of course dozens, literally dozens of UTRGV and Texas State students and staff who have picked hundreds of samples, many thousands of hours under the microscope, finding these tiny snails and working on them. So we thank all these people for making this work possible. Okay, so let me tell you about this aquifer where we're talking about. So this is in Central and West Texas and extending down into Coahuila. This is a karstic region, and it is the most diverse, arguably the most diverse subterranean fauna in the world. So hugely rich fauna, including the snails. It's also the primary source of water, drinking water for all of the people, 5 million people in Austin and San Antonio. So here is San Antonio in this region, Bexar County, and then up a little bit northeast is Travis County. That's where Austin is. And this is the sole source of drinking water for those people. So this is an area that's rich biologically and also of great human importance. Now the snails, just to give you the big picture, there's about 30 species in this aquifer system so far described, and 18 of them endemic to Texas. None of them are federally listed, although several have been candidates or petitioned for listing. About half of them or so are endangered, threatened, vulnerable, and then a handful are probably extinct, some in Mexico. If we think about where exactly they are underground, when I say subterranean, maybe you picture something like this, a cave pool. Only one of them has actually been described from a cave pool from Longhorn Caverns, Phreatic plana. More often, they're probably going to be in places like these little cracks in the rock, as you see on this cutaway view of the aquifer. So where you have in this karstic habitat, we might have snails in these little channels in between the groundwater pools. And then we also are searching for them primarily where the river surface water intersects with the groundwater. And so where you have gravels connecting the groundwater to the surface water, that's called the hyporheic zone. And that's where we're conducting a lot of our sampling in order to have access to what's normally would only find further below ground. If we look at where these are probably living, this is a large conduit, so there's a diver here at the end. That may be part of their habitat. Probably most of the little conduits and channels through the rock are going to be these dime and quarter-sized pores through the rock, and those will be lined with microbial mats, and that's probably where our snails are living most of the time. If you think about who they're living with, what else lives underground? The energy source, the basis for the food web in this system, is mostly these microbial mats. So this is the autochthonous energy generation, where you have freshwater and saltwater interface. There you can have lithoautotrophic bacteria be making energy by using minerals. And then we also have, mostly in different parts of the aquifer, have some material from above ground being washed down into the aquifer whenever there's rain. So these are the two energy sources for this group of organisms. The snails are primary consumers, so they're living in this microbial mat and eating it, along with a couple other crustaceans. They might be eaten by some additional consumers, so some other crustacean species. Nixticbromus, including some blind beetles. And then all of those would also be eaten by flatworms, blind salamanders, blind catfish, and then of course there's some things that eat all of those. So we have a very long food web for a subterranean system. We think because of the chemolithoautotrophy, it makes it such an relatively energy rich for a subterranean system. Okay, so now I want to tell you about how we go about finding these organisms. And because access to the deep aquifer is limited and challenging, we can't always get precisely spaced sampling. So we do the best that we can to understand the ranges of these organisms. So for example, we may sample a spring. So right here you have spring water flowing out from underground. And so we put a net to try and capture as much of the spring flow as we can. That's a 100 micron mesh net, so catches everything that washes out. There's a little pipe here, bit of PVC that gives a refuge, some place for the animals to hide out until we come and retrieve the net 24 hours or 48 hours later. We may do some cave exploration and explore those cave pools that you might have been picturing earlier. But mostly we're sampling the hypereic zone. So I told you the hypereic is the region of gravels between the surface water and the groundwater. So in this little stream, we are pounding a spike, essentially installing a short-term well, pounding a small spike down into the hypereic zone, and then we attach a pump to the top of the spike and pump water from below ground through a mesh net, and then wash that net down and preserve it in alcohol. Then that sample is brought back to the lab, and the debris, gravel, animals, everything is processed by hand under the microscope by, like I said, a small army of students. One of these samples could take anywhere from hours to 100 hours to process, depending on how many animals are in it and how challenging the substrate is to work with. And so my students in my lab spend a lot of time just like this. And here's some of the organisms that they might find. Some snails, some mites, some worms. Now, let's take a look at some of what we find in the gastropods. So we'll talk about Phreatodrobia a couple of slides because this is the biggest group. These are mostly narrow-range endemics. So for example, Phreatodrobia punctata is known from just a couple of sites, and this edge here is called the Balcones Escarpment, and there's a lot of springs that are right along this edge. Punctata is known from a few. Conica, it has a wider range, and then Phreatodrobia imitata is only known from 2 wells that are adjacent to each other, and these are 2 deep, relatively salty wells, so they're getting down towards the salt water. We've looked in the hypereic zone in this area, and we can't find them in more shallow environments, so they appear to be restricted to deep wells, just these 2 so far. Recently, we have discovered and named 3 new species in Phreatodrobia. So Phreatodrobia embossa, we just described from Carolyn Springs in the west. Phreatodrobia bulla from Williamson County. And we'll think about the shape of bulla, this conical shape, because we'll look back at them in a moment. And then Phreatodrobia spica, which has a relatively wide range, all the way from the Devil's River over through the eastern part of the aquifer. Now there's also 5 species of Phreatodrobia that are flat, that have a depressed shape. These are mostly narrow-range endemics. Phreatodrobia plana and rotunda are found in the same habitats in Comal Springs and San Marcos Springs. We recently expanded the range of rotunda by maybe 100 miles. And then Phreatodrobia candelensis was just very recently described from Candela in Coahuila, the only Mexican member of this genus. Now, Phreatodrobia nugax and micra are a special case. So micra is here on the left and shown with the white dots. Nugax, all of these shapes here on the right are ascribed to Phreatodrobia nugax, and those are the black dots. So their ranges overlap quite a bit. I'd say, well, that's not entirely necessarily accurate because they're really hard to distinguish. They're probably both widespread. When they're together, you can kind of tell them apart, but when they're alone, they look the same. So I would say the taxonomy is very rough on these right now, and they're going to need some work to disentangle. Whenever we sequence nugax, they pop up in about 4 different parts of the tree. And so one of our next big tasks is going to be tracking that down and really trying to figure out where nugax is and what else needs to be described. Now, if we switch to a different genus, Balconorbis, these are also relatively depressed snails. This was a monotypic genus for a long time. Balconorbis uvaldensis was described from the Uvalde area in the western part of the aquifer. And then recently, we've moved what used to be Phreatodrobia coronae into Balcanorbis. This species is interesting because it has a couple of cool dimorphic features. So for example, this individual on the left is depressed and has these big rib shape structures, and the individual on the right is globose with no ribs. Some of them are completely smooth in terms of the big ribbing. They both have microsculpture. So they tend to be dimorphic in these two features. And then this new species of Balcanorbis is called Balcanorbis sabinensis, and so it was just described from the Rio Sabinas, one of those regions in Mexico that has some of this similar fauna, related fauna. Also, a group of genus that's entirely in Mexico are the group of Coelics. So Landii and Hubsii are both found in Cuatro Ciénegas. And then Perezensi is in the Rio Sabinas, and then this little area of springs called Viesca. And these are the only sites that are known for these. Another endemic Mexican genus is Phreatomascogos, and these are known from very narrow ranges. Garcia saceroi is known from Candela, and then Gregoi is just known from the Rio Sabinas. These were just described though, so maybe we can expand their ranges with additional sampling. One last group of Mexican genera, Paludiscala caramba, is from Cuatro Ciénegas, and then two, Phreatoviesca and Spinopurgus, are both from this Viesca Springs area in Coahuila, and from my best recollection, both of them are believed to be recently extinct as this spring system has gone dry. However, if they are truly phreatic, they may still persist underground depending on the groundwater level in the region, but it would take some more sampling and maybe sampling the hyporheic zone in that area to see if we could dig them up. Okay, so if we come back to Texas with one last group, Stigopurgus bartonensis was described maybe 40 years ago from Barton Springs in Austin, and then we have added about 20 more sites and greatly expanded its range. We just recently added 2 more species to Stigopurgus. Stigopurgus variabilis. This is both in 2 individuals of variabilis, all from the lower canyons of the Rio Grande, and then Stigopurgus gracilis from a little tributary of the Rio Grande. Finally, Texapurgus diaboli. We just synonymized Trionia diaboli and Texapurgus longlei as the same species, and these are just found in the Devil's River. Now, our sampling the last 2 years has really been driven by our observation that when we lay out all of our samples and the distributions of all these snails, we have some big gaps in our sampling. There's big chunks of the state we hadn't covered. And so we propose to fill in the gaps. And so our previous sampling are these little black dots, and our current sampling are shown in white. So we really were trying to fill in our understanding of the snails around the Frio, the Nueces, the Medina rivers, and then a little bit some sites up in the Concho. We've so far sampled over the last 2 years, 76 sites, 192 samples, and we're so close to being done processing those. We have less than 10 less still to sample. But some of the things that we found so far. Vitropurgus we described last year from just one site, Comal Springs in Central Texas, and our sampling over the last 2 years has actually added more than 10 new localities. So now we have a much broader range for this species. We've also found something fun about Phreaticeras. So Phreaticeras taylori was described from the Frio Nueces River area, and you can see that here in this part of the tree, the top part of the tree. That's the central dots, cluster of dots. Well, what we've been calling Phreaticeras taylori from Bell County over here on the eastern part of Texas is sister to Phreatodrobia bulla. So what we've realized is that we have multiple evolutions of an uncoiled trumpet-shaped snail in this area. And this snail allegedly goes all the way down to Cuatro Ciénegas, so we'll have some work to do sorting out the names of Phreaticeras. We've also described several potentially new species, with all of these being new morphologies that we hadn't previously seen, including another uncoiled snail. We see lots of populations of things that are halfway uncoiled or where the aperture is pulling away from the body whorl. And so I'm curious to see how all of those are related. Then finally, our current work is to take all of these new samples and do some genome skimming to identify our new species, see if we get a better resolved phylogeny to understand these convergent morphologies, and help resolve some of these really troublesome species. And we're also trying to address some questions about gene flow. We've sampled sites on and off the aquifer to understand the influence of the hyporheic zone on gene flow. All right, just to wrap up, I want to tell you we just got some additional funding to sort out the Phreatodrobia nugax versus Phreatodrobia micra question using genotyping by sequencing data. And so I'm looking for a master student who'd be excited to take on the challenge of working with some of these tiny snails and go see some cool habitats and neat springs in Texas. So I look forward to addressing your questions. Thank you for listening to my talk.